So that was, I could confirm that. That was all fine. And the next breakthrough really came from going to a seminar. It was a wonderful talk by this guy here, John Gerhardt, who described experiments that he and his colleagues had been doing on frog oocytes. Oocytes, by the way, are the cells which are going to be eggs when they grow up. And in frogs, it's the hormone progesterone that turns an oocyte into an egg. And I'll show you, this is not the world's most dramatic movie. Watch carefully about there on that oocyte. There we go. I've added some progesterone. And a white spot very slowly appears. It takes about 5 to 7 hours, depending on the temperature, for that white spot to appear. Not, you might think, the most dramatic things. But were you to look inside this oocyte, which has now become an egg, what you would see, actually, is chromosomes again, lined up on a second meiotic spindle. And what a Japanese postdoc had discovered a few years before in working in Yale University was what progesterone did was to activate an inactive precursor of an enzyme which somehow catalyzed cell division. Unfortunately, what was this substance? Don't know, because every time Gerhardt and his colleagues, and he was a very expert protein chemist and enzymologist, the thing would disappear. It was terribly, terribly unstable, so they couldn't find out what it was. But it quite clearly had some very interesting properties. First of all, the idea that there could be an enzyme that catalyzed cell division struck me as amazing, because most enzymes, to be honest, aren't terribly interesting. I mean, they break down your food, or they catalyze the addition of a phosphate onto glucose, or things like that. They're often quite difficult to assay. But the idea that there could be an enzyme that catalyzed cell division, I thought this was a really delicious problem, and I wanted to know more about it. And down the line, Gerhardt and his colleagues discovered that this magic factor, which they called MPF, the maturation promoting factor, but you could equally well call it mitosis or M-phase promoting factor, always found when cells were in metaphase, the time when the chromosomes are lined up, never found while DNA is being synthesized, not only present in unfertilized oocytes, but also in dividing eggs. But again, in the eggs, only when they were at the height, the peak of cell division. And you might say, well, who cares about frogs? But a group of, again, another Japanese group working in starfish found that starfish had MPF. And amazingly, starfish MPF would work in frogs. And eventually it was found that if you took metaphase-arrested human cells, they contained MPF, which would make, we could assay in a frog. So the pressure to find out what MPF was sort of intensified at that point. It seemed to be a universal catalyst of cell division. And at this point, I did my experiment. Now, I should explain that I did this experiment really to I wasn't looking for anything, but let me show you what I saw. In fact, what I was trying to do was to compare patterns of protein synthesis in parthenogenetically activated eggs with properly fertilized eggs. And I think this was because I'd had a religious upbringing, and the idea of working on the molecular biology of virgin birth somehow appealed to my subconscious. And this is what I saw. It's a very simple experiment. You take some fertilized sea urchin eggs, you add some radioactive label and radioactive amino acid, and you just take samples every 10 minutes or so. And I think what you can see, I'm used to reading these things, lots of people of course aren't, and basically that's a protein, that's a protein, that's a protein. They're all radioactive. Now what you would expect is that because there's radioactivity present all the time, is they just get, these bands get stronger and stronger, blacker and blacker as time went past. And what I saw was that this protein here, which is actually a very strong protein early on, gets to be the strongest protein at here about an hour, and then faded away. And then it came back again, then it faded away. And I could easily go on to show that every time the cells divided, this protein went away. It's a bit easier to see. This is a different organism. This is a clam. And here you can really see, now you couldn't see the other protein in the sea urchin, but in the clam it's absolutely clear. There are two of these things called A and B. Here's a control, which is just getting blacker and blacker with time. And here these two things are bopping up and down. So I thought, oh my goodness me. that is absolutely amazing. It doesn't explain entry into mitosis, but the disappearance very naturally and easily explains the end of mitosis. Now, I didn't dare to hope that I had stumbled on MPF, although its behavior was remarkably reminiscent, because I thought, well, it can't be MPF, actually, because this protein accumulates steadily like that, whereas the cells enter mitosis rather abruptly. And so there's not very far from perfect. It turns out that was something that one shouldn't have worried about. But from being an expert in protein synthesis, I then turned to be a beginner in the study of the cell cycle. So I started to read about the cell cycle, and luckily for me, almost nothing was known about the control of the cell cycle in those days. And what was known was mostly due to these two gentlemen, my fellow laureates, Lee Hartwell, who was the real pioneer, and Paul Nurse. And what they'd done was to isolate mutants in yeast which failed to divide and identify the genes which were responsible for catalyzing cell division. So very much like MPF, but a genetic approach. And they came to focus on one particular gene, which in Paul's organism was called CDC2, and that's the nomenclature that we generally use today. But Lee's enzyme was CDC28, and it looked like this, and it was an enzyme. It's called a protein kinase. It's an enzyme that puts phosphates onto other proteins. But if you made this stuff, and that was quite easy to do, it had absolutely zero activity. And what was funny was that there was I called my protein cyclin, partly because it went up and down, and partly because I was a very keen cyclist at the time, so it was a sort of fun name. And the end of the story, which took us, we were really slow, and let's not go there. Turned out that actually cyclin was the activating subunit of Lee and Paul's enzyme. It was as simple as that. So CDC2 plus cyclin equals MPF. This is the active form of the enzyme. And how it works is really, really simple. Cyclin accumulates, turns on CDC2, phosphorylates lots of proteins, and the cell enters mitosis. Cyclin is degraded, CDC2 turns off, and the cell exits mitosis. The reason this was a really important discovery was at the time, people would have said it was quite impossible that one protein could go away like that. We now know that actually it's very possible, and it's often used as a control mechanism. So now we understand what controls cell division. Aren't we in a very good position to study what goes wrong in uncontrolled cell division, such as what we find in cancer? And as you can see, this sailor, which I found in the St. Bartholomew's Medical Museum, probably smoked one too many clay pipes in his lifetime, and has got a horrid tumor of his lip. And you can see, although it's a horrid tumor of his lip, unfortunately, the tumor cells know only too well how to get themselves a blood supply. So tumors are not only made of tumor cells, they're also made of perfectly normal cells. Otherwise, they couldn't grow like that. But you can also tell that these cells have completely forgotten that they were supposed to be a lip, and they are growing out of control. And so when I say we, my colleagues have looked at what goes wrong in tumor cells, and it turns out that what goes wrong is that they get mistakes in their chromosomes, and one can now take these tumor cells, sequence their chromosomes, and find out what's wrong with them. And this is a quotation from one of the pioneers of this, Mike Stratton, who runs the Wellcome Trust Center in Hinkston. Cancers arise owing to mutations in a subset of genes that confer growth advantage. Now remember, actually, cancers arise, when you mutate genes, they generally lose activity. So how does losing an activity cause you to grow out of control? Well, I guess it must be that you lose inhibitors of growth. We thought that systematic sequence of cancer genes would lead to the discovery of additional cancer genes, fine. But what they discovered was a rather horrifying fact. They discovered that a typical cancer cell had no fewer than 25,000 mutations in it. Now that provides a problem because it's very difficult to say which of those 25,000 is actually responsible for the growth of the tumor. And if you look at cancer chromosomes, you can see these chromosomes, if they were normal, all ought to be the same color, and you can see this sort of extraordinary. These chromosomes have broken and chromosomes have rejoined. There are abnormal numbers of chromosomes, so there's too much of some proteins. Other proteins have been broken in half and rejoined to other proteins. I mean, it's a complete mess. And it would be very difficult to say, whether it's that little blob there or this funny chromosome here, which seems to have 1, 2, 3, 4 chromosomes that have broken and rejoined to give this giant abnormal chromosome. What actually causes the tumor to grow out of control? Well, it's hard to say. But what people do think is, and they say, is that cancer is a disease of the cell cycle. And I'm now an expert in the control of the cell cycle. But of course, you have to be a little bit careful here, because we know what happens if you inhibit the cell cycle completely. That was tragically discovered among the victims of the first atomic bombs. I mean, if you have a lethal dose of ionizing radiation, which blocks all cell division in the body, you last about a week and you die an agonizing and very unpleasant death, because we all rely on growth and replacement of cells in our body all the time. I mean, you know that red cells are constantly replaced because they constantly decay and go. So it's no good inhibiting. But would it be possible? Do cancers have an Achilles heel? And the focus was very much on this. This is not my protein, but it's the same idea. Cyclin D and CDK4 and 6 clearly have a role in what people call getting out of the cell cycle. Most of the cells in your body are most of the time actually resting. They're not dividing. Yet, if you cut your skin, as you know, suddenly the cells at the margin of the cut will leap to life and grow and divide and make good to repair the cut. So cells are sort of sitting there with their antennae twitching all the time. And it turns out that it's this protein here which is part of that antenna. We don't still know all the details. So people very early on, when they discovered this, they say, no good inhibiting these guys because this controls mitosis, and without mitosis you can't replace cells. No good inhibiting him because he controls entry into S phase, and if you can't, that will just be like standing next to an atom bomb when it goes on. But maybe if you make inhibitors against these, we could somehow find that the tumor cells had got something wrong. They were a little bit trigger happy. They would divide too easily when they shouldn't divide. And so drug companies made inhibitors, and in 2005 it was announced that a potent and selective inhibitor of cyclin-dependent kinase 4 and 6, they're very closely related by the way, had been discovered. But when the original researchers looked at whether it was going to be any good and show any potency against tumor cells, they found that it didn't. It inhibited normal cells. And it turns out the only reason they came to this conclusion was because they only looked after one day. And the great Dennis Slayman, who was very much involved in the development of the drug Herceptin, an antibody that's helpful in certain forms of breast cancer, decided to take a second look at this, and instead of growing the cells for one day, grow them for 3 days. And there he found that actually the tumor cells seemed to be quite sensitive to this drug, and so initiated a trial, and you won't be able to read this, I'm afraid. The addition of paclitaxel, which is the name of the drug, to letrozole, which is another standardly used drug for breast cancer, in this phase 2, significantly improved progression-free survival in women with advanced estrogen receptor-positive and HER2-negative breast cancer. So in fact, CDK inhibitors are proving to be useful. I don't know why, but they are, and that's great news. Absolutely great news. I mean, who knows? And of course, we now have to find out exactly why the cancer cells are more susceptible to than their normal counterparts. And who can say where the road will lead next?